Go.
Integration is for preparation for the cell divisions, and prophase is the nuclearly disappear and the chromatin condenses into chromosomes. The nuclear envelope breaks down, and mitotic spindle is assembled. Then is the metaphase. Metaphase is when chromosomes are aligned across the metaphase plate, and microtubules pull each chromosomes apart into two chromatids. Next stage is anaphase. The microtubules connected to the chromatids, shortened as tubulin units, are uncoupled at their chromosome ends, and overlapping microtubules interact to push the poles farther apart. The last phase is the telophase. Nuclear envelope is restored, chromosomes disappear into chromatin, and nuclearly reappear. Then there is a cytokinase. Cytokinase occurs which divides the cytoplasm to form two cells. In plant cell, a cell plate forms, while in animal cells, cleavage flow forms.
So meiosis is the process of cell division for germ cells and can be used for sexual reproduction. And so first in meiosis, they use homologous chromosomes, and it's kind of like this, where there's two sets of chromosomes, one from the maternal and one from the paternal. As you can see, this. So these are the chromosomes that are used during meiosis. And first in interphase, pairs of homologous chromosomes in diploid parent cell are duplicated and become homologous chromosomes. As you can see, there's two homologous chromosomes, and a total of 4 sister chromatids. And then in meiosis 1, homologous chromosomes separate, and in meiosis 2, the sister chromatids separate. And so in meiosis 1, it's a separate of homologous chromosomes, and first, before this interphase, chromosomes are replicated, and in prophase 1, synapsis occurs where homologous chromosomes pair up, and it forms tetrads, which consists of 4 sister chromatids. And crossing over also occurs at the chiasmata in prophase 1, which allows for genetic variation. And then in metaphase 1, the tetrads line up, and in anaphase 1, pairs of homologous chromosomes separate, and the sister chromosomes here are still attached by the centromere. And then in telophase 1 and cytokinesis, 2 haploid cells are formed, and each chromosome have 2 sister chromatids. And then meiosis 2 occurs, and in meiosis 2, no interphase occurs, but in prophase 2, there's no crossing over, and the spindle fiber forms. And in metaphase 2, chromosomes line up. In anaphase 2, sister chromatids are separated, and in telophase 2, 4 haploid cells are formed, and the nuclei reappear, and each daughter cell are genetically unique. And also in metaphase 1, there is the independent assortment that takes place, which allows for genetic variation. Go.
Mitosis and meiosis share the same similarities. First, basic steps are similar: prophase, metaphase, anaphase, telophase, and cytokinase. Produce new cells, start with single parent cell, and go through interphase before.
So I'll talk about the differences between mitosis and meiosis. And one of the ways meiosis is different from mitosis is that meiosis produces genetic variation, and this happens by synapsis and crossing over in prophase 1, and also in metaphase 1, pairs of homologous chromosomes line up on the metaphase plate, and in anaphase 1, homologous pairs separate, and sister chromatids are still attached at the centromere. So the basic differences are that mitosis occurs with somatic cells, while meiosis occurs with gametes. And mitosis goes through 1 division, while meiosis goes through 2 division. And mitosis produces 2 diploid daughter cells, while meiosis produces 4 haploid daughter cells. And mitosis, the cells produced they have no genetic variation, are clones of each other, while meiosis are genetically different, and it can be like 1 in 8 million alike. And mitosis goes from zygote to death, while meiosis goes from female before birth, and it matures on and on. And The main difference could also be that the purpose of mitosis is growth and repair, while the purpose of meiosis is reproduction. And meiosis has the genetic variation that occurs through crossing over independent assortment and random fertilization.